Welcome grade 12. I want to welcome you to the Junior Tikkie Winter School. There will be a series of videos which will be live streamed in the next few weeks on the juniortikkie.online platform. Please use the given material as revision and also look at all the grade 12 content for life sciences on our platform. So let's get started. I am Ms. Kutsia, and today we will look at the Life Sciences Paper 2 that was written in November 2021. So when we move to the contextual questions, one will most likely then find an image. So before you read the questions, it is very important for you to examine the given diagram or the image. So in this case, you should recognize that we have a DNA molecule. So why do I say that this is a DNA molecule? So firstly, if you read the first sentence, it says, the diagram below represents part of a DNA molecule. So there's my first statement that indicates that this diagram has a DNA molecule in. Secondly, you will notice two phosphate sugar backbones, or it will indicate two DNA strands. So if you look at the image, you will see that they also indicated on the image that there's a strand 1 and a strand 2. This does not mean that they will necessarily always indicate that there's a strand 1 and a strand 2. So if you go back to previous question papers from previous years, you will notice that there will be cases where they don't indicate strand 1 and strand 2. Then thirdly, you will also notice base pairing that takes place between the nitrogen base pairs, and this will only be found in a DNA molecule. So before we work on how to read the questions, let's quickly have a look at the short summary that I made of the DNA structure. So let's look at the summary on the DNA structure. So firstly, DNA has a double helix structure. So very important for this is you have to indicate that it's a double helix. If you just indicate that it is a helix, you will not receive your mark. So it's a double helix structure or a double stranded structure. So what are the monomeres of a DNA molecule? So the monomeres are the building blocks of the DNA molecule. So it's like building a house. When I'm building a house, the brick will represent the monomere of the house because it is the building block. So remember when to ask, if we ask what is the monomere of the DNA molecule, you have to indicate that it's a DNA nucleotide. So it is very important to write the word DNA before the word nucleotide. So if we look at the picture at the bottom, it represents the structure of a nucleotide. So firstly, we have a deoxyribose sugar. And then we also have in the left top hand corner, you guys will see the small little circle, which is known as the phosphate group. And then we will move over to the nitrogen base. So we have 4 nitrogen bases. We have adenine, guanine, cytosine, and thymine. So in a DNA molecule, we have nitrogen base pairing that takes place. So during base pairing, we will have adenine that will always form a bond with thymine and guanine that will form a bond with cytosine. So that is what a nitrogen base pair is, so that we can form a double-stranded molecule. So the bond that forms between these nitrogen bases is known as a weak hydrogen bond. So let's move on to how to read my questions. So to read the first sentence is extremely important to understand what the question is about. A lot of the learners I've noticed over the years that just feel that there's no important information that could be given in the sentence that's given at the top of a diagram. But in this case, they give a very important part of the information where they tell you that it's a DNA molecule. So they say the diagram below represents a part of the DNA molecule, and that will already make it much easier to answer the questions. So the first question is a very simple question, and this is a normal, you have to know that if you know the answer, you know you studied it. So there's no trick to this question. It's to identify certain structures. So the first structure that you have to identify is molecule X, then sugar Y, and then the bond W, so that's the bond that forms between the two nitrogen bases. So then in question 1.4.2, they ask you to give the collective name of parts X, Y, and Z. So collective name refers to one word, one name that we use for the three different structures together. Then the next question states that it says that you have to state the natural shape of the DNA molecule. So this one is very important because we will all tend and we all want to say that it is a helix structure. But again, remember from my summary, it is very, very important to indicate that it is a double helix structure. The next one is that we have to name a process where DNA makes an exact copy of itself. It literally goes and it makes a copy of itself. Then question 1.4.5, they say name 2 places in an animal cell where DNA is located. So it's very important that it's 2 places that you mention, because if you only mention one, you're going to lose a mark, and that it's specifically only in an animal cell. Let's move on to the marking guidelines. So this is the memo that gives the answers to the questions. It will also indicate where the mark will be given. So for my first question, where you had to identify molecule X, the answer is that if we look at the image, it indicates a nitrogen base pair. So in this case, you cannot necessarily answer which nitrogen base it is, but if you know your structures quite well, you can indicate that it's cytosine and guanine. Then the next one was that you had to identify sugar Y. So here it is very, very important that you give the name of the sugar. Because this is a DNA molecule, it is deoxyribose sugar. So you will look at the marker location and you will see that they give it for deoxyribose, not for sugar, because the word sugar is already indicated in the question. So it's very important that you indicate that it's a deoxyribose sugar. Then the bond that forms, which is W, so this is the hydrogen bond. It's a weak hydrogen bond, but you will see they only give it here for the naming that it is a hydrogen bond that forms between the two nitrogen bases. And also again, not for the word bond. Then question 1.4.2, they ask you to give the collective name, and that's a nucleotide. So it is very, very important. In this case, they did not indicate that it's a DNA nucleotide, but if you go back to previous question papers, they always indicate that it's a DNA nucleotide. So please write DNA nucleotide, rather be safe than sorry. So it's a DNA nucleotide. In this case, they already indicated that this image is a DNA molecule. So that's why they did not add the word DNA to this question. But to teach yourself this, rather add DNA nucleotide, because in the next question paper, you do not know what they would ask. If they include the DNA molecule to tell you that it is one or not, you have no idea. Then they ask for you to state the natural shape of the DNA molecule. So this is a double helix. Again, if you just write double-stranded or if you write helix, you will not be given the mark. Then the process where DNA makes the exact copy of itself, that is DNA replication. Here they give you the benefit of the doubt, where if you just write replication, they also rewarded the mark. Again, rather be safe than sorry and include DNA replication. I know that they have moved to new examiners, so people that's setting the question paper, and there has been a few changes that we've noticed last year in comparison to the previous years. So question 1.4.5, name 2 places in an animal cell where DNA is located. So here you can indicate that it's in the nucleus or in the mitochondria. They also give you the benefit of the doubt if you write in the chromosomes, chromatin network, nucleoplasm, or even chromatids. So the trick here is that it's an animal cell. We've taught you that you find DNA in the nucleus, the mitochondria, and the chloroplast. But it's very, very important that you do not write chloroplast here, because that is only found in the plant. And that's the trick to this question, because a lot of learners will write chloroplast. If you write nucleus, chloroplast, and mitochondria, you have mentioned the 2 places where I find DNA in the animal cell. But in a question like this, they only mark the first 2. So even though mitochondria was mentioned, you mentioned in this 3rd position, so I only mark number 1 and number 2. So let's move on to question 1.5. So again, we are represented with an image. So before we read our questions, we have to examine our image to understand the questions that's being asked before we can answer the questions. So when we look at the given image, we will read the first sentence, which is really important. So they say the diagram below represents a chromosome pair undergoing a process during meiosis. They do not indicate if it's meiosis 1 or it is meiosis 2. But if you know meiosis well, you will immediately be able to identify the phase that's taking place here. So this phase specifically is prophase I. So I'll go through the summary, how you will know that it's prophase I. But firstly, we have homologous chromosomes. Crossing over has not taken place, so you will see that one chromosome is indicated by white and the one is indicated by black. The reason why I use different colors is just so that if crossing over takes place, you can see which section belongs to which chromosome. So they are exactly the same. So the color does not indicate that they are different. So let's look at the summary of chromosomes and prophase I. So firstly, to look at the chromosomes that we will find during prophase I or during meiosis, we call them homologous chromosomes. So these are 2 chromosomes that will be the exact same shape and size. So these chromosomes will always have the same alleles at the same locus. So that means we will find the same gene at the same location on these chromosomes. One of these chromosomes will come from the dad and the other chromosome comes from the mother. So during prophase I, we have a process that takes place that's called crossing over. So the reason for crossing over is to ensure genetic variation, because this is extremely important for survival. The position where crossing over takes place is called chiasma. So this is very, very important to remember for exam. So if they ask what process takes place, it is called crossing over. And if I ask what do I call this area or this position, we refer to it as chiasma. And then in the last diagram, you will notice that these 2 chromosomes have switched sections of the chromosomes, which indicates that crossing over already took place. So if we just quickly look at the structures of the chromosomes, you will see half of the chromosome will indicate and show that it's a chromatid, and then the center of the chromosome is known as the centromere. So let's read the questions of 1.5. So again, we start with the first sentence at the top of the diagram because it might hold important information. So the diagram below represents a chromosome pair. So this will indicate that it's a homologous pair of chromosomes undergoing a process in meiosis. So then by looking at the diagram, you need to see that the 2 chromosomes are 2 different colors, they are the same shape and size, and that they are crossing over. As soon as you see that there's a point where they cross over, it is prophase I. So, question 1.5.1 is name the following. So, what they indicate here is name the organ in the male. So, it's a human male where meiosis takes place. Then, name the process represented in the diagram. So, you will see that the process, so if I said in the summary, if it's the process, it's called crossing over. If they ask you identify area or location P, it will be chiasma. And that's how you will be able to differentiate between each other. Then question 1.5.2 is very simple because it's just labeling. So, this is something you just had to study. So, area P, so you will see that it's indicated with a dotted line, so making a circle around the area, structure Q, and structure R. So let's have a look at the memo. So question 1.5.1, they asked us to name the organ of the male human body, or the organ where meiosis takes place in the human male, and that would be the testes. If they asked the female, it would have been the ovaries. Then they also asked the process that takes place, and that's crossing over because they indicated and they asked the process that takes place. If we look at question 1.5.2a, they ask you area P, so that's chiasma. Then structure Q is the centromere, and then also we have the chromatid. The reason why it's a chromatid and not a chromosome, because structure R, the label indicates only half of the chromosome. So it does not indicate the entire chromosome. So that's very important. If you wrote the word chromosome here, you would not have received your marks. So the last question for question 1 is question 1.6. So when you look at this image, it is going to give you quite a scare because it is a bit everywhere. So this is known as a dihybrid cross. So luckily, we will never expect from you to ever do this cross on your own. So what we will do is we will give you the table or the Punnett square with the cross already done, and we will then leave out certain genotypes and ask you to fill it, or then give us the phenotype of a certain genotype. So you must still understand what is going on in this picture. So what we normally use here, it's a normal complete dominance cross where one allele is dominant and the other one is recessive, but the trick here is that we don't use one trait, but we use two. So let's quickly, before we move on, look at the summary of a dihybrid cross. So here is where a little bit of the math comes in, where we use the FOIL rule, how to get the big Punnett square. Again, you do not need to know this. So this is just for interest's sake to understand. So what FOIL means is the first will then bond or be together with the outside, and then we will move on to the second one where it says, let me just wait for that one, where the inside then goes with the last one. And that's just to show you how the gametes will form. So how I usually do it is I allow each of the gametes to form, or each of the alleles to go together to form a gamete. It's a little bit easier, and I'll show you something different to the FOIL rule that I use, but it comes down to the same thing. Because it's the first and the outside and the inside and the last, it scrambles my brain a bit. So to make it a bit easier for myself, I'll quickly show you guys how mine is done. So what I do is there will be an indication like the big H, so the capital H will go with S, and the big capital H will go with the small s. And then the small lowercase h will go with the big S, and the lowercase h will go with the small s. And I'll do exactly the same for the genotype on the right hand side, where the lowercase h will go with the small, and then the lowercase again with the small, and then the second lowercase h will then go with the small, lowercase s, the first one, and also it will go with the small s, the second one as well. So that's how I do it. It works exactly the same. That is just the method that works for my brain, because FOIL can get quite confusing. And they use this in peer math. So it might not be known for everyone, but they have taught this in grade 9. So you should have a background on this, but grade 9 is quite a while ago. So A dihybrid cross refers to two traits or two characteristics that we will look at once, because the word or the prefix di refers to two, where the monohybrid was much easier because it was only one characteristic. So in this genetic cross, we looked at the shape of the pea, so is it round or wrinkly? And then also we looked at the color of the pea, which is yellow and green. So again, we looked at two characteristics at once. So let's read question 1.6, and again, the first sentence is extremely important for you to understand this question, especially a dihybrid cross. So let's start. They say, in humans, short fingers are capital letter F, and a widow's peak is capital letter H, and they are dominant over long fingers and a continuous hairline. So what is widow's peak? So a continuous hairline, if you look at the image on the right hand side, you will notice a continuous hairline in the younger adult, where it has a straight line where we have the hairline. So it's a continuous hairline. And as this person gets older, they then show signs of widow's peak, where it sort of forms a M, where the hairline becomes thinner. So we had a man and a woman that decided to have children, and both of these parents are heterozygous. So what heterozygous means is that the alleles are different to one another. So if we take short fingers, so short fingers is a capital letter F, but the father has short fingers, but it is heterozygous. So it means it's a capital letter F and a lowercase f. If we look at widow's peak for the father and the mother, because both of them are heterozygous, they will carry an allele that's capital letter H and a lowercase h, because they indicate there that they are both heterozygous. If you miss this clue, you will have the entire thing wrong. So then they give you the table. They already did the cross for you, and they will never expect you to do this cross, and they will leave out certain parts of it. In this case, you will just notice Z that was left out in the table, and then you just have to fill the missing space or the open space. So in question 1.6.1, they state that they wanted the genotype of Z. So what you are looking at in the table, if we look at the first column, it says, or the second column, and my second row, they say capital letter F, capital H, capital H, and that's a genotype. So it's your genetic makeup for these alleles. The phenotype will be what will I see in the offspring, what will be visible. That's the phenotype. So here they ask the genotype of Z, and then you just have to put the genes or the genetic, the gametes together to form the genotype. Then they ask to give the genotype of the parents. So here you will get 2 marks for one answer, because the two parents are exactly the same. At the top they mentioned and said that both parents are heterozygous for the two traits. So then they ask the number of genotypes that could result in the offspring with short fingers and a continuous hairline. So the trick here is that they gave you the phenotype. So the easiest to approach this question is to quickly write down the possible genotypes for the phenotype that was given to us in question 1.6.2b. Then the alleles for a continuous hairline. So what the allele would be for a continuous hairline. So in question 1.6.2, they ask you for the phenotype of the child who is homozygous for both of the recessive alleles. So homozygous again refers to alleles that is the same. So it's going to be the phenotype of the child of the recessive alleles. So again, short fingers and widow's peak is dominant. So they just want the phenotype for the recessive alleles. So that's going to be for long fingers and a continuous hairline. But the trick is that they asked the phenotype, not the genotype. So let's quickly have a look at the marking guidelines. So question 1.6.1, they wanted the genotype at Z. So my answer will be small f, and capital letter H and a lowercase h for this genotype. So how we got it, you will move to the left-hand side of the table and you will see that you have in row number 3, you will have a small f and a capital letter H. And if you move back to Z and to the top of the table, you will see that there is a small f and a capital letter H. And then you group the letters together where your capital letter will always be first. Then question number 1.6.2, they ask you for the genotype of the parents. So they said that the parents are heterozygous for both of the alleles. So that means it's capital letter F, capital letter H, and a small h. And you get 2 marks for one answer because it was both parents are exactly the same. Then they ask you the number of genotypes that could result in the offspring with short fingers and a continuous hairline. So here you just write down the specific genotypes for short fingers and a continuous hairline. And then if you go and count these genotypes, you will see that there's only 3 offspring that will have this specific genotype. Then the allele for the hairline. So not the genotype, not the phenotype, only the allele, and that's a lowercase h. And they like asking this, and that's the trick, because you want to tend to give the genotype, which is small h, but they only ask the allele. Then they ask the phenotype of children that's homozygous for both characteristics. So if it's homozygous and it's recessive, so it's long fingers and a continuous hairline. And this is also 2 marks for the one answer. So you have to have long fingers and a continuous hairline for you to obtain both of these 2 questions. Thank you, grade 12s. Please make sure to watch the next few videos on the question papers.